Hello everyone, welcome to Biomix Sub. In this today's video, we will discuss about some of the previous year question of cell cycle regulations which are asked in CSIR Life Science exam. By discussing these questions, we will also discuss about the concepts. So let's get started with the question. The first question is, following statements were made about cell cycle regulation in eukaryotes. ये जो present है, ये कुछ statements दिया गया है eukaryotes के cell cycle के regulation के बारे में. The first statement tells that activity of maturation promoting factor MPF rises and falls in synchronously with the concentration of cyclin B. ये जो बोल रहा है, ये जो cyclin B जो present है, it is in the M phase और ये it is being synchronously regulated by the maturation promoting factor. This is the correct option. Okay. Next is a CDC25 phosphatase mediates removal of phosphate from the inhibitory tyrosine residue Y15 to yield high active MPF. So during the cell cycle regulation, there are both inhibitory kinase present as well as some of the phosphatase are also required. Okay, there is a kinase which is known as V1 kinase. Okay, what this V1 kinase does is it helps in the phosphorylation of threonine 14 as well as tyrosine 15. Okay, so the work of the CDC25 phosphatase is it will remove the phosphate, mainly the inhibitory phosphate like threonine 14 and tyrosine 15, and it will help in the activation of the MPF. Okay, so this B option is also the correct statement. The next statement. MPF specifically phosphorylates and depolymerizes lamine A and C, but not lamine B. This is the incorrect option because it is depolymerizing both lamine A, lamine C as well as lamine B. Like here you can see this is the MPF, cyclin and CTK of M phase. What does it? It helps in chromatin condensation by phosphorylation of condensins and cohesin. Apart from that, it is also required during nuclear envelope breakdown. How this nuclear envelope breakdown is taking place? It is by the phosphorylation of lamines, that means lamine A, lamine B as well as lamine C. It is by the nuclear pore complexes, phosphorylation of nuclear pore complexes as well as inner nuclear membrane proteins. Okay. The next work is fragmentation of Golgi apparatus. It phosphorylates and help in the fragmentation of Golgi apparatus. Where it phosphorylates the Golgi matrix proteins. Okay. Next is spindle formation. It helps in the phosphorylation of centromeres. Kinetochores as well as microtubule associated protein for spindle formation. So this option is the incorrect option because it will help in the phosphorylation of the lamine B. As C is the incorrect option, we can eliminate this A, C and D. Okay. Now let us see the D statement. MPF phosphorylates H1 protein. Yes, this MPF which is present, it is phosphorylating the H1 histone. Okay. Next, in cytosaccharomyces probe, overexpression of V1 protein decreases length of G2 phase and extends the period of M phase by functioning as a mediator of MPF activity. So what happens? What it is telling, when the V1 protein, what was this V1 protein? The V1 protein, it is a inhibitory kinase. Okay, it is a inhibitory kinase where it phosphorylates, as it is a kinase, it phosphorylates the threonine and tyrosine, threonine 14 and tyrosine 15. So what it is telling that, the statement is telling that when this V1 is overexpressed, then it decreases the length of G2. How it will decrease the length of G2? When it is overexpressed, it will phosphorylate this two amino acids and it will help in increasing the G2 phase, not decreasing the G2 phase. Okay, so this is the incorrect option. Now let's move towards the next question. The following statements were made about cell cycle regulation in fission yeast. ये जो statements कुछ present है, it is of the regulation of the fission yeast. यहाँ पे क्या बोला गया है? CDC25 जो है, what is the CDC25? CDC25. So before solving, let us see this diagram. So here there is M-cycline and CDK1. The CDK and M-cycline, they are inactive. They are in the inactive form. Okay, so for its during the processes, what happens, there is a kinase which is known as V1. What does kinase do? It helps in the phosphorylation of the inhibitory. It is a inhibitory kinase, so it phosphorylates some of the portions like threonine 14 and tyrosine 15. Next, another activating kinase comes, which is known as CDK kinase. Okay, CDK activating kinase, which is known as CAK. So where it phosphorylates in threonine 161 position. Okay, when it gets the proper time, when it needs to be turned into the mitotic phase, during this time what happens, the CDC25, which is a phosphatase, what it does, it helps in removal of the inhibitory phosphate, that is threonine 14 and tyrosine 15. Then it enters into the M phase, or the M-CDK is activated. Okay, so now let us try to solve this question. A CDC25 mutant cannot enter mitosis due to its inability to remove the inhibitory phosphate. Okay, a CDC25, why the CDC25 was present? Because it is a inhibitory protein. So what is does it help in, so ये जो sorry the CDC25 which is present, it is a phosphatase. What it does, it helps in removing the inhibitory phosphate. So when this is mutated, when this CDC25 is mutated, what it does, it cannot enter into the mitosis phase because it cannot do its work. So this cell cycle cannot enter into the mitosis phase. That is the reason A is the correct option. Okay, next, V1 consistently maintain the CDK in an active state to maintain cell size during cell cycle. Does this V1 consistently maintains the CDK? No, it is not consistently maintained. It is not consistently maintaining the CDK in its active state. It is helping in the phosphorylation of the inhibitory amino acid.